Very pleased to have you here, and you've got an amazing event coming up soon. Tell me a little bit about TAT. It's happening in March in Paris.
So TAT is Targeted Anticancer Therapy. It's certainly one of my favorite meetings of the year. I've been going for a number of years, and I now have the honor of serving on the scientific organizing committee. TAT is a meeting that's mostly focused on new targets and new drugs, but it really goes beyond that. It covers the spectrum of biology to target validation to phase 1 clinical trials, and it's kind of the launching pad to phase 2 studies or even phase 3 studies. One of the advantages of TAT is that I'd say it's relatively small meetings, 300 to 500 people, and you get to interact with the attendees. So as a more senior person, I like to interact with some of the younger folks to talk to them about the history of the targeted therapy and how we have arrived to where we are today.
And you yourself interact with many people in the cancer business, because you're both a clinician and a translational scientist, and you work with ASCO, you work with the NCI and with SWOG. So based on your experience, what do you think TAT will achieve that other conferences might not be achieving?
Well, I think it's the personal interactions. I mean, you have an outstanding agenda, you have discussions and great speakers on emerging targets, and you not only talk about the drugs hitting those targets, but it's the biology behind that, because we need to establish, we need to establish a framework where we understand the biology, we identify biomarkers, we can identify subsets of patients likely to respond to therapy, and then design the clinical trials.
One thing that clinicians like is the collection of good guidelines, so why do they need to get their hands dirty and really understand all of the scientific nuances?
Well, the guidelines are the last step. In other words, that's once you have approval of a drug, then you have to set guidelines so that the cancer community knows what's appropriate or perhaps what's not. At TAT, it's more the early development of the drugs and the biomarkers, and it's chemistry among the participants. You have enough senior people where they can mentor the younger people and like I said, tell them the history of targeted therapy and where we've made mistakes in the past and how we can do better in the future. The younger people have a chance to interact with some of the more senior folks to learn, but they also get to interact with people from the pharmaceutical industry, some basic biologists who will be attending the meeting, and other investigators who they will collaborate with in order to achieve their goals and hopefully help our patients.
What's in it for the clinician?
A tremendous amount. You know, when you go to some of these meetings, you don't always get a full background on the biology behind the target. And at TAT, you have not only wonderful clinical investigators, but also the translational investigators who may have spent their whole career, 20 or 30 years, investigating a particular pathway in cancer. And these are the folks who are at the plenary session at TAT and who are lecturing and who know the target and the pathway inside and out. They know the pitfalls, they know the biomarkers, and truly it's a credit to the folks who organized TAT to be able to attract some of the best speakers, not only on both sides of the Atlantic, but also from other areas of the world, such as the Far East, other parts of Asia, etc.
Could you give me a few hot tips about what's going to be exciting in terms of some of these processes that look promising?
You know, obviously immunotherapy is very hot, and we all go to the lectures, including here at ESMO. I've attended the immunotherapy lectures. I think what we need to learn about is what are the best biomarkers for selecting patients for immunotherapy, and what are the best combination treatments, whether it's two immunologic agents or whether it's an immunotherapy with some other targeted agent. And my area has always been angiogenesis and VEGF biology, and that seems to be a hot area. So we will not only talk about individual pathways or drugs, but perhaps the best way to combine these drugs in order to improve our clinical trials. And again, the most important thing is the outcomes for our patients.
There's some really promising pathways occasionally simply don't bear fruit. What would you say to clinicians about that sort of thing?
Well, you never know what the outcome is until you actually do the clinical trial. However, I will give TAT credit for opening up the podium to alternative ideas. For instance, just to address the point that you just raised, sometimes things look promising but don't pan out. Last year I gave a lecture at TAT to talk about the value of preclinical data. Sometimes it's very good, sometimes it is not so good, and sometimes people just select the best data to present and ignore the negative data. And by me giving this lecture at TAT and sending the message, we need to hear all the data, we need to hear the truth about both the positive studies and negative studies, and I think these are the things that TAT encourages. It's a free exchange of information, sometimes not always positive, but it's important to learn from the negative data, and we have that opportunity at the TAT meeting.
So could you sum up in a few words why people should go to TAT, both the scientists and the doctors?
Well, I think it's cutting edge science that's presented, and if I wasn't on the scientific organizing committee, I would go every year because it gives me a chance to sit in one session where I don't have to run from session to session, learn the things that I really want to learn, but it also exposes me to other areas that perhaps aren't right in my field, but things that I need to be aware of. And in the field of oncology, I just mentioned an example of 2 different targeted agents targeting 2 different pathways that may seem to have a biologic rationale when we treat our patients. So I think you get a broad view of the emerging targeted therapies, understanding the pathways behind it, so that when the clinical trials are done, you're not just given a set of drugs and a set of slides, but you're actually understanding the pathways so that you can help design the clinical trial, implement the clinical trial, understand the potential toxicities, and improve our clinical trial system.